Imagine a medical mystery so baffling it turns the brain into a sponge, is inevitably fatal, and is caused by something, well, incredibly unexpected.
It does sound like something out of science fiction, doesn't it? But it's very real.
Yeah, it's not a movie plot. It's a genuine, perplexing challenge that medicine grapples with.
Indeed. These are among medicine's most unique and, frankly, confounding conditions. And that's precisely what we're untangling today here on The Deep Dive.
Exactly. We're diving into a stack of reliable medical sources to understand these rare, neurodegenerative conditions known as prion diseases.
Our mission for you, especially if you're, say, a first-year medical student trying to get your head around this, is to equip you with the essentials.
Right. What prions are, how these diseases show up, they're really bizarre ways of occurring and transmitting, and what we can actually do.
And we'll use some helpful anchors, maybe a few vivid metaphors, and pose a couple of questions to really help nail this down.
Okay, let's kick off with one of those questions then. Ponder this. How can a protein, just a protein, a basic building block of life, become an infectious agent?
It's a fundamental question that really shook biology. Keep that in the back of your mind. We'll definitely circle back to it.
All right. So keeping that unsettling image of a sponge-like brain in mind, what are these things? What is a prion?
Okay, so prion itself stands for proteinaceous infectious particle. And the key word there really is proteinaceous.
Meaning it's made of protein, no virus, no bacteria.
No fungus, nothing like that, just protein. It's quite unique in the world of infectious agents.
And this discovery was huge, right? Completely changed how we thought about infection.
Absolutely revolutionary. You have to remember, before Stanley Prusner's work, and he won the Nobel Prize for it in '97, the idea of something infectious without genetic material like DNA or RNA was almost unthinkable. Heresy, really.
Wow. Okay. And these diseases, they have an acronym.
Yes, TSEs. It stands for Transmissible Spongiform Encephalopathies.
Let's break that down. Transmissible.
Means it can be passed on.
Spongiform.
Leads to that sponge-like appearance in the brain we mentioned. Microscopic holes.
And encephalopathy.
Simply means a disease of the brain. So a transmissible brain disease that makes the brain look like a sponge. Pretty descriptive.
And that spongiform part really highlights how destructive they are. They're neurodegenerative.
Correct. They progressively destroy brain tissue. Neurons die off, leaving those gaps.
Maybe a metaphor would help here.
Yeah.
You mentioned trying to make this stick for students.
Yeah, a good one is to think of prions as, well, zombie proteins.
Zombie proteins. Okay, I like that.
They aren't alive like a virus or bacterium, no DNA or RNA, remember, but they have this uncanny ability to convert normal, healthy proteins into their own abnormal, disease-causing shape.
Like a zombie bite that turns others into zombies.
Exactly like that, but for proteins. It sets off a chain reaction, spreading this infection through the brain until the whole system is just overwhelmed by these misfolded proteins.
Okay, that zombie protein idea is pretty vivid. So these things, they affect different species.
They do. Prion diseases are found in mammals, including us humans, but also various animals.
So in humans, what are the main ones we should know about?
Well, the most commonly discussed is Creutzfeldt-Jakob disease, CJD. Then there's a variant form, vCJD, which we'll definitely talk more about.
Okay.
And there are others, like Kuru, which has a fascinating tragic history, and some rarer inherited forms like fatal familial insomnia, or FFI, and Gerstmann-Straussler-Scheinken syndrome, GSS.
FFI and GSS.
Got it. Those sound pretty severe just from the names.
They are incredibly rare, but yes, devastating when they occur.
And in animals. I think most people have heard of one famous example.
Absolutely. Scrapie in sheep is a classic one, but known for centuries. Then there's bovine spongiform encephalopathy, BSE, that's mad cow disease.
Right.
And also chronic wasting disease, CWD, which affects deer, elk, and moose. It's becoming a bigger concern in North America.
Okay, so a range of species. What does it actually look like when someone, a human, gets one of these diseases? What are the signs? It's a really difficult picture. Symptoms often start subtly, maybe with anxiety or depression, but then they progress, often quite rapidly.
How so?
Patients develop ataxia, that's a profound loss of physical coordination, difficulty walking, controlling movements. Memory loss and cognitive decline are usually severe.
So problems thinking and remembering.
Exactly. And often muscle spasms, what we call dystonia. And in later stages, incontinence is common. It's a very debilitating decline.
It sounds incredibly grim. And the prognosis?
The prognosis is, unfortunately, uniformly grim. These diseases are, without exception, inevitably fatal.
No cure at all.
No known cure. We have treatments that can help manage some symptoms, provide comfort care, but nothing to stop or reverse the underlying disease process.
How quickly does it progress, usually? With CJD, for instance.
Most people with the common form of CJD, the sporadic type, die within a year of their symptoms starting, often from secondary complications like pneumonia because they become so debilitated.
Just a year?
Yeah.
That's incredibly fast and devastating for the families.
It truly is. The speed and severity are heartbreaking.
So let's go back to the brain itself. What's actually happening microscopically to cause all this? You mentioned the sponge-like look.
Right, so that spongiform appearance comes from widespread neuronal death. The nerve cells are dying off, leaving these holes or vacuoles.
Okay.
We also see something called gliosis, which is basically the brain's support cells, like astrocytes and microglia, becoming overactive. They're trying to clean up the mess, essentially.
The brain's immune response kicking in.
Sort of, yes. But it's not enough, and might even contribute to the damage in some ways. And importantly, you get a buildup of these abnormal prion proteins forming clumps called amyloid plaques.
Amyloid plaques, like you see in other neurodegenerative diseases like Alzheimer's.
Exactly. It's a common feature in many protein misfolding disorders, though the specific protein involved is different. In prion disease, it's the PrPSc protein forming these plaques.
Okay, so it's a combination of neurons dying, support cells reacting, and this junk protein building up. Quite a destructive process.
Extremely destructive.
Now, you mentioned how rare these are. Something like a couple of deaths per million per year for the common type.
That's right for sporadic CJD. They are very uncommon, thankfully.
But what's really fascinating, and maybe unique, is how they occur. Our sources highlighted 3 distinct ways.
Yes, this is one of the truly remarkable things about prion diseases in medicine. They can happen sporadically, be inherited genetically, or be acquired through transmission.
Right, and you had an anchor for us, for the students listening.
Yes, a simple way to remember these 3 mechanisms is the anchor SGA.
SGA.
S for sporadic, G for genetic, and A for acquired.
SGA. Okay, that's helpful.
Yeah.
Sporadic, genetic, acquired. It really does underline how weird these diseases are arising seemingly from nowhere or from family history or like an infection.
Precisely. That versatility is almost unheard of. It makes them a sort of grim medical marvel.
So let's break down SGA. First, sporadic. That's the most common.
By far. About 85 to 90% of all cases are sporadic.
And sporadic means?
It means it appears seemingly out of the blue. No known genetic link, no known exposure to prions. It just happens.
Any idea why?
The exact cause isn't certain. The thinking is it could be a random error. Maybe a spontaneous mutation occurs in a single brain cell's PrPSc gene. That's the gene coding for the prion protein.
A somatic mutation, just in that one cell.
Possibly. Or perhaps a normal PrPC protein molecule just spontaneously misfolds into the dangerous PrPSc form, again, just by chance. A very unlucky molecular accident.
And this tends to happen later in life.
Yes, the typical age of onset for sporadic CJD is usually between 60 and 65.
Okay, so that's sporadic. Next in SGA is genetic.
Right. Genetic or familial forms account for maybe 10 to 15% of cases.
And these are inherited.
Yes, these are linked directly to inherited mutations in that same PrPSc gene. If you inherit one of these specific mutations, your risk of developing a prion disease is much, much higher.
Because it present differently.
Often the symptoms start a bit earlier than in sporadic cases, maybe in the early 50s on average, and the specific disease might be FFI or GSS, depending on the mutation, not just CJD.
And how do these mutations cause the disease?
Well, the mutations seem to do one of 2 things, essentially. They might make the normal PrPC protein structurally unstable, making it more likely to misfold.
Okay.
Or they might actually make the abnormal PrPSc shape more energetically favorable, sort of tipping the balance and making that conversion process more likely to happen.
So the gene itself creates a predisposition, makes the protein wobblier, or the bad form stickier, metaphorically speaking.
That's a good way to put it. It primes the system for failure, you could say.
Okay. And the last one in SGA, acquired. This is the rarest.
Yes, acquired forms are the least common, maybe 2 to 5% of cases overall, but they're incredibly important conceptually.
Because this is where the infectious part really comes in, right? This gets back to our first question. How can a protein be infectious?
Exactly. It's through these acquired routes transmission from an external source that we see this bizarre infectious nature of prions demonstrated most clearly.
So to understand that, we need to dig into the protein itself. You mentioned the two forms, PrPC and PrPSc.
Right. The absolute core of prion disease is this transformation. The normal, healthy cellular prion protein, PrPC,
C for cellular, the good guy.
converting into the abnormal pathogenic form, PrPSc.
SE for scrapie, the bad guy.
Precisely. Now, let's compare them. PrPC, the normal one, has a structure rich in alpha-helices. It usually exists as a single molecule, a monomer.
Okay.
And importantly, it's protease sensitive. That means our body's natural enzymes, proteases, can easily break it down and recycle it. It does its job, often at the synapse between nerve cells, and then gets cleared away normally.
Seems pretty standard for a protein. Now the bad guy, PrPSc.
Very different. PrPSc has a structure dominated by beta sheets. This different folding causes it to clump together to aggregate.
Like those amyloid plaques you mentioned.
Right. Exactly. And crucially, this beta sheet structure makes it highly protease resistant. Our enzymes find it incredibly difficult to break down.
So it just hangs around and accumulates.
It does. And this PrPSc form is the one associated with infectivity. It's the zombie form that can convert others, and it's notoriously hard to get rid of, even with harsh sterilization methods.
Okay, maybe time for another metaphor. You mentioned Legos earlier.
Yeah, let's try that. Imagine normal PrPC is like a perfectly shaped Lego brick. It fits neatly, does its job in building structures.
Okay.
PrPSc is like a misshapen, sticky Lego brick. It doesn't fit properly. It can't build anything useful. Worse still, when it bumps into a normal Lego brick, PrPC, it somehow forces that normal brick to also become misshapen and sticky.
So one bad brick corrupts the others.
That's the idea. It acts as a template or a seed. The PrPSc molecule makes contact with a normal PrPC molecule and induces it to refold into the abnormal PrPSc shape.
And then you have two bad bricks.
And those two can convert two more, then you have 4, then 8, 16. It's a chain reaction, an exponential amplification of the misfolded pathogenic protein.
Wow, that templating mechanism sounds insidious. It just keeps recruiting normal proteins to the dark side.
It's the heart of the disease process, and because the PrPSc is resistant to breakdown, it just accumulates and accumulates.
Leading to the neurodegeneration we talked about, the cell death, the sponginess.
Right. The accumulating PrPSc is directly toxic to neurons, cytotoxic. Plus, you might be losing the normal function of PrPC as it gets converted. Some evidence suggests PrPC has antioxidant roles, so losing it could make cells more vulnerable.
Okay, and as the neurons die and get cleared away by those microglia cells,
They leave behind the gaps, the vacuoles, creating that characteristic spongiform pathology, and the PrPSc aggregates form the plaques.
it paints a pretty clear, albeit grim, picture of the pathology.
It does, which leads us back to that second question we wanted you to think about.
Right. If this PrPSc is so dangerous, and it can be acquired, say, by eating contaminated material, how does it actually get from your gut all the way up to your brain to start this mess?
It's a crucial question for understanding transmission. How does it travel? Let's explore that by looking at some historical examples.
Good idea. That question, how prions travel from the gut to the brain, it seems like understanding past outbreaks would shed light on that.
Absolutely. History provides some stark, tragic lessons. The classic example is Kuru.
Kuru in New Guinea, related to cannibalism.
Yes, specifically ritualistic endocannibalism, practiced primarily by the Fore people, where they consumed the brains of deceased relatives. It led to a devastating epidemic of a prion disease very similar to CJD.
And stopping the practice stopped the epidemic.
Eventually, yes. It was definitive proof that prion disease could be transmitted through ingestion, through eating infected tissue. A truly grim but vital piece of the puzzle.
And then there's a more recent and perhaps more widely known link
Between BSE, bovine spongiform encephalopathy or mad cow disease, and variant CJD vCJD in humans.
Right. BSE emerged in UK cattle in the mid-80s.
1986 is when it was first officially noted, yeah. And initially, there was significant denial about the potential risk to humans.
I remember the images. Wasn't there a government minister famously feeding his daughter a burger on TV to show it was safe?
That was John Gummer in 1990. In retrospect, it's quite a jarring image, knowing what we know now. It really highlighted the initial underestimation of the risk.
And then people did start getting sick.
Yes, the first deaths from what would be identified as vCJD occurred in 1995. There were 3 that year. The peak was around 1999-2000.
And how many people were affected in the end?
In the UK, there were 123 definite and 55 probable deaths attributed to vCJD. The last recorded death from it was in 2016.
And vCJD was different from the usual sporadic CJD, wasn't wasn't it? Affected younger people.
Dramatically different in that respect. The median age of onset for vCJD was only 26.5 years old, and the median age at death was 28, compared to sporadic CJD hitting people in their 60s.
That's chilling. So these examples prove ingestion is a route. What other ways can people acquire prion disease?
Besides ingestion, the other main route is iatrogenic transmission.
Meaning through medical procedures.
Exactly. Tragically, there have been documented cases from contaminated corneal transplants, duramater grafts, that's the tough membrane covering the brain, and even from injections of human-derived growth hormone prepared from pooled pituitary glands before synthetic versions were available.
Wow, so contaminated tissues or instruments were transmitting the prions.
Yes, which really underscores how persistent these prions are.
So back to the travel question, ALF to brain. How does it work? If you eat it, it doesn't just get digested.
Apparently not easily. Remember, PrPSc is protease resistant, so after ingestion, it seems to survive the digestive tract to some extent.
Okay, then what?
It's thought that specialized cells in the gut lining, perhaps M cells overlying Peyer's patches, which are part of the gut's immune system, take up the prions.
And then the immune system gets involved.
Yes, the prions then appear to replicate, or at least accumulate within lymphoid tissues, particularly involving cells called follicular dendritic cells. The spleen and lymph nodes can become reservoirs.
Okay, so it's amplifying in the immune system first. How does it jump to the brain?
The current understanding is that it travels from these lymphoid tissues along peripheral nerves, particularly nerves of the autonomic nervous system that supply organs like the spleen and gut. It's like it hijacks the nervous system's own wiring to slowly creep up towards the spinal cord and ultimately the brain.
A slow, stealthy invasion via the nerves.
Yeah.
That makes sense for the long incubation periods we often see.
Exactly. It can take years, even decades, for symptoms to appear after exposure.
And all this reinforces the idea that it really is just a protein, right? No virus needed for this transmission.
The evidence is overwhelming. You can take brain tissue from a sick animal, inject it into a healthy one, and transmit the disease. But extensive searches have never found a causative virus or bacterium.
And you mentioned UV light.
Right. Treatments that typically destroy nucleic acids, like UV radiation or certain nucleases, don't reduce the infectivity of prion-containing material. But treatments that denature or destroy proteins do reduce infectivity. It all points back to the protein.
Which brings us to a huge practical problem. If these things are so tough and infectious, how do you manage them in, say, a hospital?
It's a massive challenge. PrPSc is incredibly resistant to standard sterilization methods. Heat, radiation, common disinfectants often aren't enough. This makes prion diseases an infection control nightmare.
So there must be special guidelines.
Oh, absolutely. Detailed government guidance exists for handling patients known or suspected to have CJD, or even those considered at increased risk due to family history or past exposures, especially if they need surgery.
What kind of precautions are we talking about?
Well, for surgery, they often try to schedule these patients at the end of the day's operating list.
Why is that?
To allow for extra, intensive cleaning procedures afterward without delaying other cases. You also need minimum personnel in the room, and everyone needs extensive PPE.
PPE-like.
We're talking liquid repellent gowns, plastic aprons over those, double gloves, masks, eye protection like goggles, or even full face visors. And ideally, for symptomatic patients, all this gear should be single-use and incinerated.
And the surgical instruments, that seems like a major risk.
Huge risk. The guidance strongly recommends using single-use disposable instruments whenever possible, and these must be destroyed by incineration afterwards, not just thrown in clinical waste.
What about instruments that have to be reused?
That's where it gets really tricky. They need to undergo specialized decontamination protocols, which are much harsher than standard sterilization. And crucially, there needs to be rigorous tracking systems in place so you know exactly which reusable instruments were used on a high-risk patient. In case that patient is later confirmed to have CJD, you might need to recall and destroy those instruments.
Wow, the logistics sound intense. What about blood safety? Especially thinking about vCJD transmission.
Yes. The potential for vCJD transmission via blood transfusion led to a whole raft of specific safety measures, particularly in the UK.
Such as?
Things like withdrawing and recalling any blood components donated by someone who later goes on to develop vCJD.
Makes sense.
Importing plasma needed for blood products from countries with no or very low BSE risk, like the USA, to reduce reliance on UK-sourced plasma.
Okay.
Implementing universal leukodepletion, that's filtering out the white blood cells from all blood donations intended for transfusion, as those cells were thought to potentially carry prions.
Right, you mentioned the immune system involvement.
Exactly. And also, a controversial but precautionary measure. Not accepting blood donations from anyone who received a blood transfusion themselves since 1980 during the main BSE vCJD risk period.
That must exclude quite a few potential donors.
It does. It highlights the better safe than sorry approach taken. And overall, there's a big push to promote the appropriate use of blood and alternatives just to minimize transfusions generally.
So lots of precautions. But in terms of actual treatment for someone who's sick, still just supportive care.
Largely, yes. Research is ongoing, exploring compounds that might interfere with the PrPC to PrPSc conversion or help clear existing PrPSc. But as of now, no effective cure or disease-modifying therapy has been established. It remains a huge unmet need.
Which brings us towards the end of our deep dive for today. It's been a, well, fascinating look into a really unusual corner of medicine.
It certainly has. We've covered a lot what prions are, these unique neurodegenerative diseases they cause, the whole SGA framework of sporadic, genetic, and acquired forms.
And how that bizarre protein-only infection actually happens through conversion and transmission.
The discovery of infectious proteins, Prusner's work, it genuinely revolutionized biology and our understanding of disease mechanisms. It opened up a whole new category of pathogen.
And maybe a final thought for our listeners, especially the students, to carry forward.
Well, think about this. We've focused on prion diseases, which are rare. But the underlying theme, protein misfolding and aggregation, is central to many much more common neurodegenerative diseases like Alzheimer's and Parkinson's.
That's a really important point. They all involve proteins going wrong and clumping up, even if they aren't transmissible in the same way prions are.
Exactly. So, while only prions have this unique transmissibility, studying these zombie proteins might hold crucial lessons for understanding those other devastating conditions. What can the mechanisms of prion replication and spread teach us about stopping protein aggregation in Alzheimer's, for instance?
Could understanding how PrPSc spreads cell to cell inform how tau or alpha-synuclein spread in other diseases?
That's the hope. This fundamental protein problem, proteins adopting toxic shapes and spreading pathology, is a massive challenge in medicine. Studying the most extreme example, prions, might unlock secrets applicable across the board. It keeps the research incredibly vital and raises questions we're still actively trying to answer.